Okay, good morning, everybody. Very happy to have you with us this week. We hope everybody's doing well and enjoying what looks like much cleaner air to breathe, and hopefully that continues. Today we have a really exciting talk. Dr. David Weinreich is going to be speaking. He's the Vice President of Regeneron. Before we get into that, we have our usual updates. I want to start with just mentioning, you may notice that many of our virtual background is a picture we did a year ago. We had this idea, I think, Bob, it might have been your idea to do a group picture of all the women faculty to celebrate Women in Medicine Month, which is every September. And we were really excited to do this group picture last year. We thought it was really successful. This year, we were wondering how we could do that. And then the idea came up to let's not let everything that's going on keep us from celebrating this month the same way. So we're planning to do a Zoom event this evening at 4 p.m. You may have seen the email. Helena is going to send out the link in about 5 to 10 minutes when we have everybody who's joined, and it'll be a reminder link that will be an email as well at 4 p.m. today. It's going to be a picture for those of you who are busy and can just hop on for the picture. Please hop on at the beginning for the picture, and there'll be some guest speakers and some other stuff going on as well. So we hope that you join us today. And I just want to say thank you very much for all our faculty and particularly our women faculty for doing what you do. And we just want to make sure that we take this opportunity to really celebrate you. So thank you so much. Hope you can join today. And we'll also be sending out the link, which has been in emails as well, for all the virtual backgrounds. Lindsay and Cassie from the communications team created some really nice virtual backgrounds to celebrate Women in Medicine Month. So please, especially if you're joining today, if you can, throw one of those up in your background as well. With that being said, Dr. Huja, as usual, thank you for the weekly updates. I'll turn it over to you for some updates.
Thank you very much. Good morning, everyone. And I just want to also acknowledge all of the work that Kathy Garzio and Rob Harrington have done for our Women in Medicine, supporting, promoting. It's been very, very transformative over the years. So thank you. I'm happy to report that today's COVID numbers are going down, as is the air quality index. So we're seeing clearer skies. Here's a heat map of the world regarding COVID, and you can see some of the hot spots. And just, looking at the top 3 with regards to numbers that have been increasing, you'll see them here. We talked about Israel last week. I also wanted to share just a quick slide about the deaths and comorbidities associated with them. And you can see here that, hypertension, diabetes do remain strongly correlated. And there may be other confounding factors that go along with that. And there's also a strong correlation with obesity, particularly as you look at the younger patients that have obesity and COVID death rates. Just looking overall at the United States, cases do continue to go down, fortunately. And within our Bay Area counties, the numbers have definitely shown a consistent decline. And then within our own hospital, the numbers trending down since about late July, early August. More granularly, we have 18 COVID patients in the hospital this morning, of which 5 are in the ICU and 3 are intubated. From March 1st to September 13th, we've had 430 at SHC. The ICU and death rates remain stable as the last few weeks. Same with the female population. The age breakdown is about the same. And as we look at the deaths by age and gender, you can see here the breakdown remains similar. There's been not much change over the past few weeks. Just a quick snapshot of the pediatric inpatient data. You can also see their numbers have gone down, fortunately. And when we look at ethnicity, gender, the Hispanic population continues to be the majority in the ethnicity range, and just slightly more males than females for the pediatric population. Looking also at chronic health conditions in the pediatric population for patients that were admitted with COVID, you can see, pulmonary or cardiac disease represented here, any other chronic medical condition. But fortunately, the majority here were previously healthy, and you have a small percentage with oncologic diseases. And then finally, for the pediatric patients, the majority were asymptomatic positives admitted for other diagnoses, about 34%, or 47 patients total for all of the admissions represented in this pie graph are shown here, 34% admitted for COVID. Finally, thank you to my data team, and I'll turn it back over to you. Thanks, Errol.
Great, Nira. Thank you so much. I appreciate that. Before we get started with the next week's presenters, I also wanted to just highlight Dr. Joy Wu. Dr. Wu, thank you so much for every week providing the summaries ever since the first grand rounds, COVID grand rounds back in March. And she puts them together every week, and then we send them out in the email, as well as she tweets them out as well. Here's one of the tweets she sent out recently. Just wanted to focus talking about Women in Medicine Month. She had actually did a little analysis and found that just about 50% or over were women faculty from presenters and panelists. So thanks for letting us know that we're doing a pretty good job there. And you know, we just have so many leaders in the whether we're focusing on it or not, which we are, we're going to have a lot of faculty coming from women because we have so many great leaders. So thank you so much for analyzing that, and thank you for doing these summaries every week. I want to also mention to you, next week we have a great talk from Dr. Robert Khalif. He's the former FDA commissioner. We're going to have a conversation with him with the COVID theme and talk more about different themes around COVID and all the medications that we're looking at seeing coming out in the future. The week after that, Dr. Michael Snyder has been working on a lot of projects around COVID. One of them is the use of wearables in diagnosing COVID early onset, and he's going to talk about the research he's been doing. And then the following week, we have doctors Shushi Anand, Julie Parsonet, and Maria Montez Rath to talk about renal involvement in COVID-19. So now I'm really excited to introduce to you David Weinreich. David Weinreich joins us all the way from Tarrytown, New York. Thank you, David, for that beautiful virtual background screen of San Francisco. And thank you so much for being with us. David's joining us from Regeneron out in New York. He's the Senior Vice President of Global Clinical Development. Before that, he was the Senior Vice President of Late Stage Clinical Development and Medical Affairs, and the Senior Vice President and Head of Global Development and Specialty Medicine at Bayer Pharmaceuticals before that. He's been in multiple other companies before, including Amgen and many other years of experience in biotech and healthcare. He got his bachelor's in computer science back in Columbia, an MBA at McDonough School of Business at Georgetown University, and then went to medical school at at Boston University School of Medicine. Dr. Weinreich, thank you again for joining us. I'm going to stop sharing my screen, and Talia, I'm going to ask you to go ahead and start sharing. Dr. Weinreich, thank you again for being with us today. We're really excited to hear more what you have to talk about in regards to monoclonal antibodies and all the work that Regeneron is doing to hopefully get us out of the pandemic as soon as possible. So I'll turn it over to you, Dr. Weinreich. Thanks so much.
It's my pleasure. Thank you very much for the invitation. So I thought I'd start a little bit just in case you don't know much about Regeneron and our history, and then delve into some of the work that we've been doing with antiviral rapid response in COVID. Could I have the next slide? So Regeneron is, I guess everyone would call us a big biotech at this point, but we have not shed our humble beginnings where science leads everything. If you look at the makeup of the senior leadership of our company, more than half have their advanced doctoral degrees, either MD, PhD, or both. Our diverse board is likewise, more than half have their MD, their PhD, or both, including several Nobel laureates, and our chair, Roy Vagelos, who I believe one of his children is on faculty at Stanford. Because we're run by scientists, it's part of our DNA, and our goal here is to utilize all of the scientific technologies that we've invented to try and help patients with a myriad of different diseases. The next slide. And we've been doing this for over 30 years. We celebrated our 30-year anniversary a little over a year ago. And I'd like to think that we're somewhat successful at doing this. It's sticking to the core principles that were true when we were a 50-person company, now as we're a little over a 9,000-person company. Next slide. We have 7 approved drugs utilizing the technologies that have been built here. All of the drug candidates were developed in-house, both from research all the way through development. We continually rank quite highly in various different employer surveys like in Science Magazine. And we are committed to things that are maybe in the very long run useful for drug discovery or patient identification, but you might have a hard time trying to put a risk benefit or an ROI around it. And one of those big endeavors more recently has been committing to sequencing more than 1 million individuals by the Regeneron Genetics Center. It's a wonderful database that we utilize both in research and in development, literally on a daily basis. Next slide. And what separates us from other companies is that science being at our core is not a tagline. It's actually a belief. We're not driven by commercial. You'll see that in the next slide when I talk about the breadth of our portfolio. We don't focus on what Wall Street is interested in short-term earnings. We don't tend to out-license our drugs. And we don't focus on what there might be efficiencies from our sales force or internal expertise. Instead, we let the science guide us to novel areas of medicine, and we're not opposed to taking on the big challenge of the unknown. And we've always got a long-term view at heart. Next slide. And that drives, despite our size, a very, very broad pipeline that, while we organize it into therapeutic areas for convenience, really spans from rare diseases like fibrodysplasia ossificans progressiva, all the way to general medicine diseases like high cholesterol, from oncology across any type of oncology, solid organ, hematologic, to being one of the pioneers to suggesting that it would be okay to stick a biologic by needle into the back of the eye for various neovascular diseases. I routinely joke that internally that if we were to come up with a new target for depression, I probably would need to hire some psychiatrists because we don't have that technology, that we don't have that experience base in-house. And then promptly the RGC discovered a target for severe early onset unipolar depression. So maybe I'll have to hire some psychiatrists in the not too distant future. Next slide. Okay, so moving a little bit on to SARS-CoV-2. Next slide. As I'm sure you all know, this virus is a single-stranded RNA virus. It is the causative agent of COVID-19. It's genetically similar to SARS, as well as several bat coronaviruses. The current thinking, and I believe this is correct, is that there's a single glycoprotein on the surface of the virus called spike, which is required for the interaction with human cells via a host receptor, ACE2, and that it is this interaction that we want to try and block with therapeutic antibodies to either prevent infection or to aid in clearance of the virus. This was first reported towards the end of last year, coming out of China, and the pandemic was officially declared by the WHO in March of this year. Next slide. There's a lot of reasons to want to try human monoclonal antibodies as part of a rapid response. Antibodies tend to have limited, if any, off-target toxicities. And unlike vaccines, antibodies provide immediate protection, and the duration can be quite long. Upwards of a month is not atypical for antiviral antibodies. In addition, we all know that eliciting an immune response to a vaccine is very variable, and there are certain populations that historically have had difficulty in mounting their own innate immune response, such as elderly or patients who are immune compromised. And utilizing not just the technologies that we've developed, but how antibodies in general are produced, you can produce and isolate fully human monoclonal antibodies, and they tend to have a very low potential for immunogenicity. Now, we've taken the tact that we want to use cocktails of multiple neutralizing, non-competing antibodies. And we believe this is important to prevent mutational escape. And I have some preclinical data I'll show a little bit later, but also some anecdotes from other monoclonal antibodies that we've created against other viruses. But what's really different about Regeneron is our investment in scientific technologies over the last 30 years have allowed us to bypass some of the traditional bottlenecks in how monoclonal antibodies are identified, scaled up, gotten ready for the clinic, produced GMP, and then produced in enough quantity to actually service the market. And we were able to do this in a much shortened time frame. Go to the next slide. So all of these technologies start with Velosa and then something, and it's been a series of investments over the last 3 decades, each building upon the prior one. And the importance of this is it's turned the identification of antibodies as well as their scale up and getting them ready for the clinic from a science problem that has to be addressed individually for each antibody, each cell line that you want to create, into more of an engineering problem. And that allows our researchers to focus on the science and not focus on, can they create an antibody? Is it going to have enough yield? Is it going to be compatible with our technologies in our manufacturing group? And it provides a large degree of similarity in the backbone between each of the different antibodies that Regeneron produces. And that provides certain understanding of what a cell line is going to produce and how it's going to, how much production it's going to be, but it also allows us to go much, much faster. Next slide. So, a typical timeline to producing an antibody can easily take a year or 2 from a researcher saying, aha, I have a really good idea. I want to go after target X, to the clinical organization saying, aha, I'm ready to go with all of the regulatory discussions and manufacturing of material and so forth. Because of all of these investments over the last couple of decades, we can collapse that 1 to 2 year time frame into 3 to 6 months. And it's something that we've done previously. This is not the first time that we've done this for COVID-19. We did this for Ebola. We've previously shown that our 3 antibody cocktail targeted against Ebola in a randomized clinical study conducted in collaboration with the WHO and Doctors Without Borders and the NIH showed superiority in preventing death in the Democratic Republic of the Congo compared to the active control, which was the standard of care at the time, of another antibody called ZMapp. We've done this for a somewhat related virus, the MERS virus, that went as far as phase 1 before the disease sort of disappeared into the woodwork and we put the antibody on the shelf. But we've also done this against respiratory syncytial virus, and that's an interesting learning that I'll get to when we talk about escape. And we deployed this same set of technologies to be able to go from, wow, there's a problem in the world that we can probably help in January, to first patient enrolled in the clinical study in June, without compromising any of the usual safeguards to ensuring that the drug hits the target the way we want it to. It's has binding efficiency, in the sub-nanomolar range. We can produce large scale of this. We've done all of our safety studies to convince global regulatory authorities to allow us to go into the clinic. Next slide. So this is just giving you an idea of sort of the specific timelines. As I said, this sort of hit our radar screen in the latter half of January. We started our immunization campaign in mice in February, isolation and screening in March. We selected our cocktail in April, and within 2 months, we were in the clinic. And this wasn't that we only picked one or two antibodies, and I'm going to show you some data in a couple of slides. We actually screened thousands of antibody pairs to try and find a cocktail that was, each individual antibody was very potent, neutralized, had to bind in the receptor binding domain of ACE2, but also did not compete with another antibody so that we could have a two antibody cocktail. And to be perfectly honest, we probably would have preferred to do 3 antibodies, but this receptor binding domain isn't all that large, and despite our initial attempts across a couple thousand potential antibodies, we couldn't find 3 that all bound at the same time without neutralizing one of the, or blocking one of the other antibodies. In the example of Ebola, the binding domain is actually quite a bit larger, and we were able to find 3 non-competing neutralizing antibodies, which is why that cocktail is actually 3 instead of 2. And despite the fact that we have found a cocktail that we are testing in the clinic, we're still evaluating more antibodies, because in the long run, much like the flu vaccine changes its makeup depending on what the prevalent strain is over time, we want to be ready if there is genetic drift in the coronavirus, that we may have other antibodies ready to go that may have a slightly different binding area that may overcome a resistance if it develops. Next slide. So I have a couple of cartoons just to highlight what we're trying to do. You've got the SARS-CoV-2 receptor binding domain of the spike protein interacting with ACE2, and it is this interaction that results in infection. Next slide. The point of a therapeutic antibody is to bind to the ACE2 receptor binding domain and prevent the virus from being able to enter the affected cell, but at the same time avoid a mutant virus from being able to sort of sneak around the antibody, because then Darwin's theory will take over and you'll have expansion of the resistant phenotype. And we've seen this before, and we've previously published with what was called Regeneron 2222, which was a single antibody targeting respiratory syncytial virus that we tested in neonates for the prevention of RSV. And in that particular case, we did select a single antibody. And that trial, a big phase 3 study that was reported out in the New England Journal a little over a year ago, what we found was over the span of, I believe it was 4 seasons, 2 in the Northern Hemisphere and 2 in the Southern Hemisphere, the drug didn't work. And it didn't work because over these 4 seasons, there was a variant of RSV that happened to have a mutation that rendered resistance to Regeneron 2222. And it became one of the more dominant serotypes for those years. Now, whether or not our antibody induced it or it simply selected for it, that's a different question. But it's a real world example that mutational escape can be a problem. And it's a reason why on a go forward basis, we prefer cocktails as opposed to single antibodies. Next slide. So it's really important that all of these antibodies meet a couple of criteria. One is that they have to block replicating virus. That's the graph on the right. They have to bind to and neutralize cells that are infected, and they have to do this at extremely low concentrations. You want antibodies that not only don't compete, but they are subnanomolar or better in their ability to neutralize and block viral replication. Next slide. And then you want to prevent the ability for escape. So let me explain a little bit what this preclinical experiment is. So you take virus and you infect it onto a 12-well plate with various different concentrations of an antibody. And since you can always administer in this preclinical experiment a sub-therapeutic concentration, there will be breakthrough. And breakthrough is highlighted by color coding the well read on here. So in this hypothetical example, 6.4 times 10 to the minus fourth was where there was breakthrough growth with that level of concentration of the antibody. You then take an aliquot from where you just broke through control with the antibody, and then you re-dilute it into a new passage. And you repeat this over and over again. So it's a forced experiment to try and induce resistance. If you look on the right-hand side, this is an example of a number of different antibodies and a number of different combinations. And what you're looking for is where the blue to red transition occurs between passage 1 and passage 2. Because you can always subdose across different passages. You don't want that break point to occur at higher and higher concentrations. That's suggestive that you're inducing escape. And in fact, the very first antibody on this list, Regeneron 10989, this one universally was found to be the most potent of all the antibodies that we had screened. And on the prior slide, it was the one all the way to the left with a IC50 or 50% neutralization that was under 10 to the minus 11th. It, however, was binding in an area of the receptor binding domain that it was impossible amongst the 3,300 that we screened to find a second antibody that didn't compete. So although this was by far and away the most potent of the antibodies we found, if you look at the first line, the first row in both passage 1 and passage 2, you got escape in one passage of the cell line. And for this reason, as well as our prior experience, we chose not to put that into the cocktail. The combination that we ultimately went forward with, which is Regeneron 10987 and 10933, as you can see, going from passage 1 to passage 2, there's essentially no difference in where that red to blue transition occurs. Technically, it's 1 to the right. The right we accept, left means that you're developing an escape. And after the publication of this science paper, we've continued to run this experiment, and now we're up to passage 7, and we still have not been able to induce escape with the combination. So while this is a preclinical experiment, and our clinical experience is not with this particular virus, it's with a different virus, it's a mounting amount of data to make you worry about trying this with a single antibody. As a matter of fact, hot off the presses, you may have seen the press release this morning from another company presenting their single antibody, and somewhere in the middle of that data release, they did comment there was a small, I believe it was a 2% increase in a viral genotype that happens to be resistant to that single antibody. So 2% isn't large, it was a small study, but it's certainly all suggestive of we should be careful with single antibodies against viral targets. Next slide. So some preclinical data. We took non-human primates, we infected that. In this experiment, we gave them the cocktail prior to giving them virus, and we were looking to see whether or not you could prevent infection. And the answer is yes, you can prevent the detection out many days after the administration of virus, placebo versus cocktail. So this suggests that if you were to give an antibody cocktail to someone who maybe was, has a high likelihood of being exposed, but wasn't exposed at the time that you started. For instance, in our household contact study, you have patients who have to quarantine with an infected individual, but they aren't PCR positive. Could you prevent them from converting? And you certainly don't have time to wait 10 to 14 days to then immediately vaccinate them. This experiment would suggest you could prevent the lateral transmission from an infected individual to an uninfected individual in that setting. Next slide. On the other hand, if you first give the virus to a monkey and then you give them the antibody cocktail, can you help them clear the virus? And again, I think the answer is yes. If you, in the pure therapeutic setting, the administrating of a neutralizing antibody cocktail can help you clear an established infection. There's also another learning here, at least in monkeys, that nasopharyngeal swabs are a lot more sensitive, both in detecting the disease as well as being able to quantify viral titers. And you'll notice even across the placebo arm, there's about a log order of difference in viral titers when you do a nasopharyngeal swab versus an oral swab. And it's one reason in all of our trials, we are doing nasopharyngeal swabs, despite the fact that I think they're universally disliked by physicians and certainly the patients that have to have the swab put up their nose. Next slide. One more. So on the basis of all of that preclinical data, one more slide, we embarked upon a very broad development plan to try and evaluate whether or not what we were seeing in the lab and what we were seeing in animals works in humans. So it's comprised at the moment of 4 different studies. One is in patients who are hospitalized because of SARS-CoV-2. And it's done in cohorts. So there are 4 cohorts in this trial based on how bad their lung function is. Either they're in the hospital, but they actually don't need oxygen, but they're there for some other reason. They're on low flow oxygen like a nasal cannula, high flow oxygen like a non-rebreather, or they're required mechanical ventilation. Each of these studies is placebo controlled. It turns out there's 2 dosing arms. So it's a 1-to-1-to-1 randomization with 390 patients per cohort. There's an outpatient study which looks at patients who are PCR positive, but do not require hospitalization, either because it was an incidental asymptomatic pickup, or they are having symptoms, but they do not require hospitalization, and it's the same 390 per cohort. We're also running a lateral transmission study. So this is the household context trial I was talking about previously. This is being conducted in collaboration with the NIAID, one of the divisions of the NIH. So here we take patients in which there's an index case who is positive, who's going home to quarantine, not going into the hospital. And you've got all their household contacts that have nowhere else to go but quarantine with them. And we're stratifying for whether or not they've already been infected and they're PCR positive, or whether they are PCR negative at baseline. And we're randomizing them 1-to-1 to a subcutaneous dose as opposed to an IV to try and look at prevention of lateral transmission, primarily in the PCR negative at baseline case. And then finally, we have a normal human volunteer study where we're testing principally safety and to get some PK data from repeated administration over time. And this will be a place where we'll be able to look at whether or not neutralizing or anti-drug antibodies might materialize, something that has not been an issue with the Regeneron portfolio. And as I've said, the history here is these antibodies are very, very similar from one product to another, but that doesn't mean you don't have to test it directly. So we're doing this in a normal human volunteer study. So there's a lot of questions that hopefully as these data sets, as these studies recruit and the data sets read out, that we hope to answer. What's the best setting to test the neutralizing antibody cocktail? Is it in patients who are in the hospital? Is it patients who are mostly asymptomatic and they're at home? Or is it in patients who, once we have a vaccine, for whatever reason, didn't get vaccinated, or the vaccine didn't work, and now they still have the disease, and you want to prevent everybody else in their household from getting it in the household prophylaxis approach? How much oxygen, how bad their lungs are, does that impact outcomes? And on the next couple of slides, I'm going to show you, we've learned quite a bit from our prior experience testing an IL-6 receptor blocker, sarilumab, in this setting, that outcomes are very, very different based upon how much oxygen you need. So there may be differences in how sick you are, whether or not this cocktail might work. What is causing the ARDS-like picture? Is it the virus? Is it the immune system's response to the virus? Or is it a mixture of the two? Clearly, sarilumab or steroids, they're going to work on the immune system component. Our cocktail is going to work, we hope will work on the virus side, but maybe you'll actually need both. And lastly, can you help clear virus faster? And this is obviously important because once you turn positive and you say go home and quarantine for 4 weeks, that has implications. What if you only have to quarantine for a week? And you can get individuals back into the economy, back to work, doing whatever their day job is. This also has potential value to the world. Next slide. So we've learned a lot about what this disease looks like from our first studies looking at sarilumab. And we've recently reported out from our US trial that two things. One, the patients who are generally requiring low amounts of oxygen, they do really well all by themselves. Patients who are requiring a lot of oxygen, they don't do nearly as well, maybe as high as a 20–25% mortality rate. And utilizing IL-6 receptor blockers may have a slight trend towards improvement in outcome, but none of the trials that have been run, either the two studies that were run with our asset, one by our collaborator Sanofi ex-US, our trial in the United States, nor the Roche data with another very comparable IL-6 blocker, tocilizumab, all three of them tell a very consistent story, which is there could potentially be a slight trend towards improvement in the very critical individuals with COVID-19, but none of the studies showed statistical significance. So whether or not this is noise or whether or not it would require a several thousand person study to show the benefit, that remains an unanswered question. But we learned a lot about how to conduct our cocktail program by analyzing this data set. Next slide. So one of the things that we learned, and this wasn't necessarily how we thought about it going into the sarilumab trial, is that baseline viral titers are higher in patients who are requiring more ventilatory support. So we didn't think this. Our going in hypothesis was patients who were in the hospital with an ARDS-like picture, who've been in the hospital for quite some period of time. This was more immune system runaway. I hate to use the term cytokine storm because my background's in oncology and that kind of means something specifically. But if you'll let me have a little bit of a liberty, a cytokine storm. And that's why we would think an IL-6 receptor blocker might work. But it turns out that in fact, there is still a lot of virus and more virus in patients who are requiring more oxygen support. So that might actually suggest that the sicker individuals in the hospital might actually be a very good place to test our antibody, not in the more mild cases. And thankfully, when we set up our program, we were testing the gamut from very, very sick to very, very mild, not knowing where the best area might be a priori. Next slide. If you dig one level a little deeper, and you take just the patients who were intubated, the patients who don't do as well amongst the intubated cohort had higher viral titers. So again, it's a post hoc analysis, but the ends are reasonably good. There were 300 plus patients in this analysis. Suggests that the patients who are doing worse might actually be a very good place to test an antibody cocktail. Next slide. So we're in the middle of this journey, not at the end. There's certainly quite a bit of data to validate that neutralizing antibodies are worth testing in humans. Those studies are ongoing. I think the way that we're looking at it, both in hospitalized patients with a variety of background clinical severity all broken up into different cohorts is a clean way to test this so that imbalances in baseline characteristics don't bias the outcome that you get on the other end. That it's likely to, in hospitalized patients, I think it's very reasonable to assume that it's a mixed cause for the respiratory dysfunction, and that approaches that address the immune system, like dexamethasone, and there was some great data that came out of the recovery study from the UK's National Health Service showing the improvement in survival with dexamethasone, might be very complementary to a neutralizing antibody cocktail. And as you may have seen, we have joined forces with the UK to test our cocktail in that same recovery study on top of steroids. But that the critical thing is more well-designed and controlled clinical studies are needed. The sarilumab example is a great hypothesis-generating idea that came out of a single-arm study. It happened to have come out of China, but that's not really the relevant point. And a lot of companies, a lot of physicians jump to, this is going to help patients, and it many months later and multiple thousand patients later, maybe it has a little benefit, but it's not overwhelming, and it just speaks to single arm experiments in this disease can be incredibly misleading. And as hard as it is to have a conversation with a patient to why a placebo-controlled study is necessary, in order to answer this question for the world, we need well-designed, controlled clinical experiments, not single-arm case series. And I'll stop there, and I'll be happy to take any questions. Thank you.
David, thanks so much for all that great stuff you just presented. We're just starting to see the questions pop up here. A lot of them pop up here. The first one was one of the first questions asked. Dr. Harlan Matlis, thanks for joining us and asking this question. It actually talks, and no surprise, a number of questions related to monoclonal antibodies. And one here states, any comments on the release in the news of the University of Pittsburgh of small molecule binding neutralizing SARS-CoV-2 antibody? States it has an antibody component. It seems interesting and promising. Is it inhaled? This is maybe a competitor, or do you have any sense of any of the other work being done out there in regards to this as well?
So I'm not familiar with this particular approach, but I really don't like the idea of competitor here, right? This is not your typical, we work at biotech or pharma and we've got competing drugs that are doing the same thing. I think you have to some extent, dump that nomenclature. We're looking for a solution for the world. And if it's not our antibody cocktail, but it's something else, we should all be rooting together. Now, that is balanced by, if you try and look at 1,000 things, you may not get an actual answer on any of them. So you have to balance those two. But I'm not rooting for or against ours versus anybody else's. We desperately need a vaccine. I think we would argue that you would want to do an antibody cocktail as opposed to a single antibody for all the reasons I went into. But if it's not our antibody cocktail for whatever reason, it's somebody else's, I'm going to be there with my pom-poms, being the cheerleader for it, because I think we're going to need a therapeutic in addition to a vaccine. They're very complementary. So I think approaching this from multiple different perspectives is the right thing to do for the world.
Great, and that's really wonderful to hear. So thanks for saying that. Dr. Culver asked a good question here. Would monoclonal antibody administration affect the safety or efficacy of a SARS-CoV-2 vaccine? Or would a vaccine to the spike protein affect safety or efficacy of the monoclonal antibody? I think that's a really interesting question.
It is. I'll hypothesize a little bit. The first hypothesis is that if the vaccine doesn't produce an antibody response against the receptor binding domain, it's probably not going to be very effective. Now, that's an assumption. I could argue that if it binds somewhere else, but it hangs over the receptor binding domain, but it doesn't actually bind there, maybe that also works, but that's a hypothesis. So if you want to develop a vaccine response against the receptor binding domain, one would theorize that a neutralizing antibody cocktail that works would interfere with the ability to create an antibody response from a vaccine. So that'd be the assumption. It may or may not be true. We have not done these tests yet, but it is something that we actually list in our IB as an unknown, and we don't want patients on our clinical trials going on to the vaccine studies. The converse is unlikely to be true. So if you've been previously vaccinated, regardless of how good or bad your immune response, giving a large dose of a neutralizing antibody should have no effect on whatever efficacy the antibodies ultimately show. It'd just be the same as if you're giving IVIG for other reasons. It's just a big slug of an additional antibody. It's just, in this case, a very specific one. So it's probably, at least theoretically, a one-way problem, not a two-way problem. But we don't know the answer yet. So that's all theory.
Gotcha. Okay, great. To all attendees, thanks. As expected, we have a lot of questions popping up. If those of you who are going to ask questions, if you don't mind taking a look at what's out there and voting up the ones that may be similar, so I can make sure those questions get asked. I'll try to get to them all. Dr. Lafayette's asking, isn't there a concern that an acute rise in immune complex with antibody treatment that might further drive immune response, complement cytokines? Maybe severe acute illness won't be the best place. Careful testing clearly needed.
So I think there's two theoretical concerns that we're looking at. This is one, and the other is antibody-dependent enhancement of infection. We've looked at both of these preclinically, and our antibody cocktail does not appear to either induce immune complex formation nor cause antibody-dependent enhancement at low concentrate or at any concentration, but the concern would be at very low concentrations at the end of the dosing window. But of course, it is something we have to look at when we're giving these to humans. I can say with now more than 1,000 patients across all the different studies, at least looking at the blinded safety data, as well as hearing the IDMC's review of the unblinded safety, it has safety concerns have not materialized to date, but it's still early going.
Gotcha. Dr. Weinreich, one thing I wanted to hit on today, and they're probably unfair questions, but we're going to ask, when are things going to be, assuming that the drugs work, and, when will things be ready? There's an anonymous question here, but one person mentions very promising treatments. How quickly could production reach levels to meet current demands? Furthermore, what are the estimates for the cost of a complete treatment course? And is Regeneron taking steps for equitable access? So there's two questions here. Assuming things are good and pass, how long will it take to get this into people's hands? And are there things being done to make sure it gets to everybody's hands?
So I'll take these in a couple of pieces, and I'll try and answer it as best I can. So obviously we recognized early on that although we could be very quick to scale up into big 10,000 liter vats of in our production capacity would be insufficient by ourselves to be able to meet the demand. Consequently, and this is all public, we struck a collaboration agreement with a very strong, scientifically driven antibody company, Roche, that also had many times the antibody capacity that we do to be able to produce our cocktail at risk, assuming it would gonna work. Now, even between Roche and ourselves, there are still going to be constraints on how much drug can be produced. Antibodies are not as simple as vaccines, and it's one reason why I'm a big proponent that we have to get a vaccine, and that the therapeutic will be, I don't want to say niched, but there'll be sections of this disease state where it will be more appropriate for an antibody, but it's not a substitute for wholesale vaccination, assuming vaccines work. As far as pricing and so forth, that none of this has been decided, and thankfully this is not my area, but I'll just say that Regeneron has a history of being fair in how we price our molecules. despite all of the advancements with Eylea that have occurred over the years, despite that we were allowed to take price increases, we haven't since the first time it was put onto the market. We do believe in properly pricing our drugs. I have no reason to doubt that we will not be doing the same thing once with this particular cocktail, but it's a little early. Right now, our main focus is, does this thing even work? And I'm crossing my fingers, but we're not there yet.
Okay, great. Thank you. A couple questions just asking about penetration. One here, do your antibodies get into lung fluids? Ken Kanagatheran asked as well something similar about effective concentrations in tissues and cells where we know the host has where the virus has a presence in the host.
So it's a really great question. We think the answer is yes. We've done some preclinical experiments on this, and the way we've calculated the doses that we are administering in the clinical studies actually are utilizing some of the preclinical experiments for penetration into infected lung to try and get a target concentration in diseased tissue high enough to be neutralizing over the entire duration of the proposed therapeutic window. And what we're hoping for is that in the treatment setting, this would be a one-dose administration, and that we would have therapeutically neutralizing concentrations in the target tissue, namely mostly the lung, out a month, which should be more than enough to clear this virus. So all of those back calculations went into the PK modeling that then selected the dose that we're trying in the clinic.
Great. So yeah, Thomas Burns was asking how many doses for one full treatment. Do you think it's going to be a short treatment? Is it going to be a long infusion?
One. For the therapeutic, we are hoping that this is going to be a one-dose infusion, hopefully over an hour, with the option for a physician to slow it down if necessary, but a one and done. Now, clearly there may be scenarios where you'll need a second dose, but we're actually not testing that in our clinic. We really believe that these doses will provide sufficient neutralization that if they clear the virus, unless you get reinfected, you'll be done with one dose. Now, in the general prophylaxis setting, not the household contacts, because there we already know that an individual who's positive will likely clear their infection in a month. So if you protect the housemate for a month, you should be sufficient. There may be scenarios where you would want to have persistent protection. An example could be a healthcare worker, where it's not that you're isolating with a single household contact, you're isolating with multiple positive contacts on a daily basis. There we're hoping to demonstrate the PK and the coverage that you could get by with a once a month subcutaneous injection. Unless you were going to get vaccinated, right? I mean, the real solution here for multiple doses would be develop your own immune response, assuming that works. But as a fallback, if either the first set of vaccines don't work, or for whatever reason, you as an individual get a vaccine and you don't mount a sufficient immune response, you could have a therapeutic cocktail that would be on a once a month cadence to try and keep neutralizing antibodies so that you would not get infected.
Gotcha. Dr. Weinreich, a question here from Dr. Barry. Is Regeneron talking with the WHO to use their incentive program for marketing to the developing world?
So I'll just say that we have had conversations with a number of groups around the globe, as evidenced by our conversation I mentioned previously with the United Kingdom. I can't go into any details about any of those other conversations, but I think there are a number of groups that recognize the science that Regeneron brings to the table, and we want to work collaboratively with like-minded scientists to test this and hopefully show that it works. But if not, then know that it doesn't work and we move on to trying to test something else.
Great. Question here. Ken has a few good questions. Next one here. Instead of IDing monoclonal antibodies from animals, is it feasible to get those from patients who've recovered from COVID-19 after a strong immune response, sequence it, and now analyze, synthesize these antibodies and develop in large scale?
So the answer to that question is yes. And in fact, we did both. In fact, the super binder I was describing previously was actually isolated from an infected individual, I believe in Singapore, and turned out to be a so-called central binder, and we couldn't find a second antibody to pair with it. But both approaches are reasonable, and we've actually been and continue to look for antibodies through both approaches.
Great. Daniel Cap asks, this is an interesting question as well, similar to the interactions between the vaccine and a monoclonal antibody. How about interactions between the monoclonal antibody and our natural antibodies we might have made after an infection, past infection?
So I don't see a reason why we would expect an interaction with any innate immune response followed by subsequent administration of a neutralizing antibody. So I don't know exactly where the individuals asking this question, why we would think there would be an interaction. It would be more of an issue of if you weren't infected and we have many fold higher titers of our neutralizing antibody, our antibody would shield the normal immune system from ever seeing the virus. So you would not develop your own immune response if you had large amounts of our antibody cocktail floating around ahead of time. So I still think this is more likely to be a one-way problem, not a bidirectional problem of administering our antibody.
Great.
But it's a question that has to be experimentally answered. These are just hypotheses I'm providing.
Sure, agreed. So it sounds like it's thought to be less of an issue. We'll see in the future. Harvey Cohen asks, given the number of patients in your trial, what are the effective sizes needed to demonstrate efficacy, especially in the sick patients?
So I'm not going to go in directly to how we've statistically sized these trials. We've taken reasonable assumptions on what we think the background placebo rate's going to be for the household context study that was designed out of the gate as a phase 3 trial. So we took an attack rate, sort of scouring the literature and having discussions with colleagues at the NIAID, and then took a reasonable assumption of what we thought would be a clinically meaningful improvement and used that to power the trial. And we built some interim analyses in case that the antibody works better than we thought it did or thought it will. In the earlier trials, those are phase 2-3 adaptive studies. So the earlier parts of the trial, the N equals 390, these are primarily on surrogates of clinical activity, virology, some biomarkers, and we plan on looking into these data sets and then sizing the phase 3s as appropriate. Now, alongside of all of that, the NHS has a design for the recovery trial that is a mortality study. It's very similar to the prior arm that was testing dexamethasone, and they have their own assumptions around what the statistical, what the background rate of mortality rate is going to be, and certain statistical assumptions, and that N works out to be several thousand, just like it worked out for the steroids. So each of these other collaborative groups have their own sort of viewpoint around the statistics, and we're working collaboratively as we start to add the Regeneron cocktail to these platform studies.
Gotcha. Okay, great. David, we're just hitting 8:59 now, and we're going to probably end no matter what with a number of unanswered questions, unfortunately, but it's always just speaks to how important a topic this specifically is. I will send, if it's okay with you, David, I'm going to send you the remaining questions and via email, and if there's any you'd like to answer, we send a follow-up email to the whole community that can see all the questions, of course, along with the video and all the content today. So we'll try to get any other unanswered questions addressed. I wanted to, so Dr. Weinreich, thank you so much for joining us today out from New York and doing all the work that you and Regeneron are doing. And we hope that the data comes out positive and this is able to be an effective treatment for all of us in the future. I also, before we wanted to close, wanted to first welcome Dr. Brian Bowman. He is taking over for occupational health, and he's been the CMO of United Healthcare Alliance, and he's taken over this huge role for us. He has agreed to join us weekly on medical grand rounds and provide updates in regards to occupational health. So with that being said, I want to thank Song, thanks so much for joining, for jumping on the video here just now. Song, Dr. Chang has been not only running his huge, amazing division, but serving as in charge of occupational health during this time, which to me just is mind-boggling. And then he's been with us every week, providing updates and just basically making sure, along with infection control, we're all staying safe. So thank you so much for everything you've been doing, and just thank you so much for sticking with us and all the hard work. That being said, I want to just put one more plug in for the Stanford Women in Health, Women in Medicine event we have today. And again, thank all our women faculty and staff for everything that you do so much. And I think we'll go ahead and close from there. Thanks so much, everybody. Have a great rest of the week.